Hello friends, Dr. Ashiq here. Today I am going to discuss a important topic for you, that is transcription. Let's go and discuss transcription. Welcome to Dr. Ashiq's Conceptuals. What is transcription? Transcription is one of the important steps in that protein synthesizing process you have learned in your genetic classes, and in transcription you can see that one DNA it is converted into RNA. Actually the data or information which is present in the DNA, it is converted into that information which is present in the RNA here. Actually, who is doing this process or how this process is occurring? You know that all the processes in our body, it is done by enzymes, and this process is also done by one enzyme here. What is this enzyme is doing? You can see that the enzyme is synthesizing the RNA, or we can call it as the polymerization of RNA. So what can you call this enzyme? This enzyme is synthesizing or polymerizing enzyme, so that this enzyme which is creating the RNA, we will be calling it as RNA polymerase. And actually you can see from where this RNA polymerase is synthesizing the RNA, you can see it depends upon the DNA molecule for the synthesis of RNA. So we will give a complete name for this enzyme. The complete name of this enzyme, it is known as a DNA dependent RNA polymerase. We'll add that word DNA dependent here. So the complete name of the enzyme is DNA dependent RNA polymerase. Actually this enzyme is having so many specialities are there. I will be telling basically 3 specialities of the enzyme in this entire video, and at the end of this video I am doing some problems to understand the concept of transcription for you. So kindly watch the entire video in full length. Okay, so let's discuss the first speciality of the enzyme, DNA dependent RNA polymerase. So that you have to understand that the synthesis of RNA, it will be occurring in only one direction by the enzyme, that is from your 5' end to 3' end. What that means, if it is synthesizing one RNA, first the 5' end will be synthesized and the synthesis will be continued towards the end, that is the 3' end. So the 3' end is only formed at the last. Actually you have to know every enzyme in the genetic, like the DNA synthesizing enzyme, RNA synthesizing enzyme, or the protein synthesis, every synthesis will be occurring in only one direction in genetics, that is from 5' end to 3' end. What is the importance of this direction in the process of transcription? You know that our DNA, it will be having 2 strands are there. One strand will be having 3' to 5' direction, and the other strand will be having the opposite direction, that is 5' to 3' direction. And you know we are synthesizing only one strand of RNA, that means we are using only one DNA strand to synthesize the RNA. Let's see how this 5' to 3' direction of synthesis of RNA is influencing which strand is becoming transcribed here. Okay, you know that a DNA molecule will be having 2 strands, and these 2 strands will be running anti-parallel to each other. What that means, if one strand is having direction 3' to 5', the opposite strand will be having the other direction, that is from 5' to 3'. And while you are studying the transcription, you know that we are synthesizing only one strand of RNA, and which of the 2 strands is used as a template for that RNA synthesis. We can find out that thing by analyzing that direction of the enzyme here. You know that the enzyme can synthesize in only one direction, that is 5' to 3' direction, so that it will be synthesizing first the 5' direction. So from which end the synthesis will be starting, you have to understand that. So here since it is synthesizing the 5' first it will be synthesizing the 5' here at the 3' end, and then that newly formed RNA molecule will be coming from here like this. Are you understanding? And it will be synthesizing toward this end, that is finally it will be forming the 3' end of the RNA here. So you can see here the RNA can be synthesized only with respect to this strand, that is having the direction of 3' to 5'. So this strand, it is known as the template strand. This strand is known as template strand. Why this strand is known as a template strand? Because our enzyme can only catalyze in this direction, that is 5' to 3', and the newly formed strand will be always in the 5' to 3' direction. So the opposite strand, which is having 3' to 5', it will be known as the template strand here, and the newly formed RNA will be complementary to this strand. You have to know that if this DNA strand is having a nitrogen base pair like adenine, 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 thymine, thymine, guanine. If it is like that, newly formed RNA strand will be complementary to that template strand here. So you know that the adenine complementary thing is actually that is thymine here, but you know that in the RNA there is no thymine here. Instead of thymine it will be uracil which will be present here. So it will be for uracil for this adenine, and another uracil for this adenine, another uracil for this adenine, and you can see that it is thymine, and guanine. For this T it will be coming, what will be coming here? Adenine will be coming, this T again an adenine will be coming, and this guanine there will be cytosine will be coming here. So this will be that newly formed RNA, and this newly formed RNA will be complementary to the template strand here. So you have understand that this strand which is having the direction 3' to 5', it is known as the template strand. So can you tell me what will be that name of the other strand which is having 5' to 3' direction? Actually this is a misnomer here. What is the misnomer here? You have to know that this strand which does not code for anything, it's known as coding strand. Actually this is the most common mistake whenever that students is having, because the students usually they will thinking like the coding strand is the one which is coding for RNA. Not at all. You have to understand that the coding strand does not code for anything, and actually the template strand is the one which codes for the new RNA. So you have to understand here, so that a newly formed RNA will be synthesized in complementary to that template strand, and that coding strand that is sitting idle without doing any work here. Okay, so I hope you are understanding this concept here. Let's discuss about the transcription process here, and you know that we are having two strands, that is 3' to 5', that is the template strand, and 5' to 3', that is your coding strand is here, and your enzyme, I will mark the E as E for enzyme, and the enzyme is coming to synthesize new RNA here. But here I'll be telling the second speciality of the enzyme is that the enzyme cannot initiate the process of transcription its own. But how the enzyme will be initiating the transcription? You have to know that there should be some specific sequence should be present at the 3' end of the template strand here. That specific sequence is found that enzyme here. That means the enzyme and the sequence is meeting together, then only the process of transcription will be initiating. And what will you call that specific sequence which is present at the 3' end of the template strand? You have to call this specific sequence as what? Promoter. Promoter sequence will be present here. Okay, so I'll be showing you that actual figure here. So there will be a promoter sequence that is present at the 3' end of the template strand. So that if the enzyme is coming and enzyme is meeting with the promoter sequence, the process of transcription will begin, and it will continue towards the other end of that DNA. And when it is reaching at the end, what should happen? You know that that process of transcription should end here. But that is the third speciality of the enzyme, is that for ending this enzyme, this E, this enzyme cannot make the end actually. So how it will be ending? For stoppage of the transcription process, there should be another sequence should be here at the end of the DNA, and this sequence here, that is known as the terminator sequence here. So you can see that in this figure there is terminator sequence will be coming at the end. So we will be calling that a promoter and terminator with respect to the template strand. So you have to understand that at the 3' end of the template strand there will be the promoter will be there, and at the 5' end of that template strand there will be the terminator will be here. So I'll be giving you a short trick to remember this thing. You have to remember like the promoter will be situated at a 3' end of the template strand. So you can see this thing here. So what I am going to do, I am going to inverse this 3 here. So it will become like a PET, pet. You have to remember it like that. What that means? That promoter is situated at the 3' end of the template strand. If you remember this thing, you can remember at any other thing, because it is a frequently asking question. So in this question they will be asking like the promoter is situated at which end of the coding strand, and terminator is situated at which end of the template strand. Repeatedly asked questions are here. So you have to remember it like P3T, and you can remember it like pet here. Okay, I hope you are understanding here. So you can see that this is the total arrangement of that transcription factory, or we can call it as the transcription site here. It will be having a template strand, it will be having a coding strand, and it will have a promoter, and it will have a terminator, and this total arrangement we will call it as a transcription unit. So which all are the components of the transcription unit? The transcription unit will be having a promoter which is situated at 3' end of the template strand, the terminator which will be situated at the 5' end of the template strand, and it will have a template strand and a coding strand. And actually the template strand and coding strand together, it will be having another name that is known as the structural gene. We'll call it as the structural gene. The template strand and coding strand are part of the structural gene here. So I'm going to show you that figure of the transcription unit from your textbook, and it will be having that promoter here. You can see it is situated at 3' end, and it is having the structural gene which is containing the template strand and the coding strand, and it will have a terminator which will be situated at the 5' end of the template strand. So this is the structure of a transcription unit. You must know this is the transcription unit here. Okay, and let's go and discuss the process of transcription. So you know that for the process of transcription, this structure should be here, and what will happen, the enzyme will be coming. What was the name of the enzyme? The enzyme is synthesizing the RNA, so we'll call it as RNA polymerase. DNA dependent RNA polymerase is coming, and it is coming and binding with the promoter sequence. You have to understand that some other factors are also needed for the process of transcription. Why? Because this enzyme and the promoter need some factor, like we will call that factor as an initiation factor for initiating the process of transcription. So I will show a more diagrammatic figure here. In this figure you can see that we are having a DNA, double stranded DNA. You can see this is the 3' to 5' strand, that is that strand here, and this is the 5' to 3' strand. This is that two-stranded, double-stranded DNA, and you can see that yellow area. What is this yellow area? Actually, this yellow area is the promoter. You can see it is marked here as a promoter, and this orange bubble, this is the RNA polymerase. It is written here, and you have to know when this RNA polymerase is coming with and joining with the promoter, this also needs some other factor that is known as the initiation factor, or it is known as sigma factor. What happens when the sigma factor, it will come and bind with the RNA polymerase, and the sigma factor will activate the RNA polymerase here. Are you understanding? So what happens initially when the RNA polymerase comes and binds with the promoter, and the sigma factor will come and bind with the RNA polymerase, and it will activate the RNA polymerase. So what will happen? This enzyme will get activated, and the process of transcription will be initiating, and this step we will call it as initiation. Okay, I hope you are understanding the initiation. And what will happen after initiation? Let's see. After initiation, this RNA polymerase enzyme, can you see this is the enzyme RNA polymerase? It will be going through the double-stranded DNA, and it will be producing a new RNA. You can see this yellow line. This yellow line, it is that newly formed RNA. You know that the first formed end will be 5' end, that 3' end will be continuously synthesized here, and you can see the sigma factor is going off from that enzyme. So this is that phase of that elongation of the RNA. We will call this phase as the elongation phase, and this elongation phase will continue till it will reach the terminator. You can see this is the terminator sequence. It is shown is in a green color here. That is a termination sequence. It is there, so that the elongation process is continuing till it reaches the terminator sequence. So let's see how the termination process is occurring. When that RNA polymerase is reaching terminator sequence, you can see this is the green terminator sequence. What will happen? Some other factor, you can see it is marked here as rho, and this rho factor is coming and binding on the RNA polymerase enzyme. Are you understanding? So what will you call this rho factor? The rho factor is otherwise known as termination factor. So when the termination factor is binding on the RNA polymerase, what will happen? This rho factor will inactivate the RNA polymerase. Are you understanding? This rho factor will inactivate the RNA polymerase, and the process of transcription will be stopping, and this step we'll call it as termination, and a newly formed RNA will be released out. Are you understanding? So this is the summary of process of transcription. I told you this is the first step, that is initiation. The RNA polymerase enzyme will come and bind with the promoter region, and a sigma factor or initiation factor will come and activate the RNA polymerase here, and this process will be beginning from here. The first step, that is known as initiation, and after the beginning, that RNA polymerase will go through your DNA sequence, that is through your template strand, and it is producing a new RNA here. So you can see that this is a 5' end and this is a 3' end will be continually synthesized here. This step, it is a second step, that is known as the elongation phase or elongation step here. Okay, and after that you can see that the enzyme is reaching that green area. What is this green area? Green area is known as the terminator. When it is reaching the terminator, there is some raw factor which is coming and binding on that enzyme, and it will inactivate the enzyme, and the process of transcription is stopping. And this phase, the third phase, it is known as the termination phase, and a newly formed RNA, it will be released out of that enzyme here. Actually, you have to know that this newly formed RNA is immature. This is immature, and it cannot participate in the process of protein synthesis. It should be processed in order to make it mature enough to participate in the protein synthesizing process. So that maturation process I will be discussing in another video. So you have to watch that video to understand the post transcriptional processing of RNA here. Now I am going to discuss some of the important MCQs which is asked from this area. During that MCQ, your concept will be more clear. So this is your first MCQ. You have to read the MCQ and you have to try to answer this thing. So the question asks that the following RNA strand was produced. They are telling, they are giving a RNA strand. It is from 5' to 3' there is a new RNA strand is given, and they are asking like which of the following DNA strand could have be the template for this RNA. We have to find out the template strand here. Okay, so you know the template strand will be having the direction from 3' to 5', and fortunately every option is giving the 3' to 5', so that we don't have to think about that thing, but you have to find out which of the following is a template strand. So you can see that this is giving adenine, 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 3 adenines together, and after that it is giving adenine, uracil and guanine here. For adenine, which will be that complementary base pair on the DNA template strand? You have to understand that for adenine it will be thymine. Thymine will be there. So there is 3 adenines are there, so there will be 3 thymine, TTT, and the next one will be AUG. For adenine I told you it will be T, and there will be uracil will be coming. Actually there is no uracil in your DNA sequence, and you have to think this uracil as a thymine. Why? Because the thymine is replaced by uracil in a RNA strand, so that it is a thymine. For thymine it will be coming, adenine will be coming. For guanine it will be coming, a cytosine will be coming. So let's go to the option. So we have find out the first 3 pairs will be TTT and TAC here. So let's go, and you can find out the D option here. It will be having first TTT, TAC. So it is the right answer for the template strand. So if this strand is acting as a template strand, then only that a given sequence of RNA will be given. I hope you are understanding. And let's discuss a very similar question. Actually the question is not changed, it is the same question it is asking, and they are giving like the following RNA strand is produced. It is the same strand which is given in the previous question, and in this question they are asking like which of the following DNA strand could have been the coding strand for this RNA. They are asking it is which of the following is a coding strand for this RNA here. You have to understand that the previous question they were asking about the template strand. Now the strand is changed, which is the coding strand in that structural gene they are asking for. First of all, you have to understand that the coding strand is having the opposite polarity of the template strand. I told you the template strand will be having 3' to 5', so that coding strand it will be having the polarity of 5' to 3' will be that coding strand polarity. So if you know this much thing, you can cancel 2 options from the list. See the first OA option, it is given like 3' to 5', and the B option it is given like 3' to 5'. So you know that this is not the polarity of the coding strand, so you can cancel these 2 options first. And you can see that there are 2 other options are given, and we have to find out which can be the coding strand. Actually I will discuss this question in a bit detail. Okay, so I will draw a transcription unit first. So in that thing I am drawing a blue strand here. It will be having the polarity of 3' to 5', and I will be drawing a green strand for you. This green strand is having the polarity of 5' to 3'. So which is coding strand, which is a template strand? You know the blue strand it will be that template strand, I am marking it as T, and the green strand it will be the coding strand here. We have discussed that thing, and we are having a newly formed RNA. So this is the newly formed RNA here. So this is an RNA strand, and the RNA will be complementary to this template strand. You know that it will be having the direction of 5' to 3' here. Okay, so let me ask you something. What will be the relationship of this newly formed RNA and this coding strand? you have to understand that RNA and this coding strand will be looking similar. Why it will be looking similar? Let us take an example and let's find out this thing. For example, I am going to take the same example which is given in that question. In the question it is given like three things, AAA and AUG. I am writing here. So if it is AAA and AUG, in the previous question we have discussed what will be the sequence in that coding strand. For A, I told you it will be thymine will be coming here, so it will be T will be coming, and AUG I told you for A it will be T, and for U it will be coming A will be coming, and for G it will be coming C will be coming. Are you understanding? Okay, if this is that sequence of that template strand, what will be the sequence in that coding strand? Let us find out the sequence in the coding strand. If it is T, what will be here coming? Here will be A will be coming. If it is another T, again another A will be coming. For this T, again A will be coming. For this TAC, what will be coming? For the T, it will be coming is A will be coming here. For A, it will be coming again T will be coming, and C it will be coming G will be coming. See, it will be AAA and ATG. What is the sequence of the RNA? RNA was having AAA and AUG. Only difference is that the T is converted into U here. So what you have to understand from here, that whatever the sequence of RNA is produced, the same sequence will be present in your coding strand also. That's why it is known as the coding strand. It will be looking similar to the RNA. That's why it is known as the coding strand. And only difference is that in RNA there will be uracil will be present, and in the DNA instead of this uracil the thymine will be present here. Okay, that is the only difference between an RNA and the coding strand. So I think this thing is very easy for you now. They are giving one RNA and they are asking what is that? What is the coding strand here? So what you have to understand, that RNA and coding strand will be very similar or equal will be there. Only difference is that the uracil will be replaced by thymine here. So let's see the options. You can see that C option here, you can see it will be looking similar to the RNA, like AAA, ATG. What was here? AAA and AUG. I told you instead of U there will be T will be coming, and you can see AGU. Here it is coming AGT. Instead of this U, here will be a T is coming, AGU become AGT, and you can see AAG is coming as AAG itself. Same thing. And what is this strand? This strand will be the coding strand in the structural gene. I hope you are understanding the difference between a coding strand and a template strand here. Let's see the final question I want to discuss here. This is the final question, and in this question they are asking something we haven't discussed yet. In eukaryotes, the question is asking, in eukaryotes there are 3 different RNA polymerase. This is a true statement, you have to understand. So actually in prokaryotic organism, in a simplest organism, there is only one RNA polymerase basically, but in eukaryote there are 3 different RNA polymerase, and they are asking the RNA polymerase which is responsible for the mRNA. Okay, so you have to understand that this is a true statement that is given in the question. In eukaryote there are 3 types of RNA polymerase are there, like RNA polymerase 1, RNA polymerase 2, RNA polymerase 3, and we are having 3 different kinds of RNAs are also, like we are having rRNA, we will be calling ribosomal RNA, which is making the ribosome. We will be having mRNA, we will be calling it as messenger RNA, and there is an RNA that is known as a tRNA or transfer RNA. We will be discussing about all these RNA in another video, and there are 3 RNA polymerase each for each individual RNA here. So how will we study this thing? I will be giving you a code for this thing. You have to study the code like RMT, you have to remember RMT 1, 2, 3. You have to remember the same thing, RMT 1, 2, 3. What that means? The RNA polymerase 1 will be for the rRNA, RNA polymerase 2 will be for the mRNA, and RNA polymerase 3 will be for the tRNA. So remember it like RMT 1, 2, 3. Okay, so you have to know. So what is the answer for this question? The question is asking which is for mRNA. For mRNA, the answer is RNA polymerase 2. So guys, I hope you are understanding what is transcription, and in next video I will be posting about the post-transcriptional changing of that RNA and the different types of RNA like mRNA, rRNA and tRNA. Kindly watch that video also, and if you are having any queries regarding to biology, please feel free to comment below in the comment box. Don't forget to subscribe my channel. Okay, we'll meet you with another video. Bye bye.